Good day, I am Jianuelle Encina. I am a second year veterinary medicine student, and my topic for this PowerPoint presentation activity in this course is about the chromosomal aberrations in pigs. So first, chromosomal aberrations are changes in chromosome structure or number. Most chromosomal aberrations are known as aneuploidies or different numbers of chromosomes other than pairs. So, like us humans, animals have identified number of chromosomes such as that cattle has 60 chromosomes, carabaos has 48 chromosomes, pigs has 38 chromosomes, and when that number is disrupted, we call that aberration in number. Now, when we say aberration in structure, in one chromosome, there could be deleted part of genes or inversions of chromosomes, and so on and so forth. Chromosomes are condensed strand of genetic materials. Chromosomes can change in size or shape. These modifications are known as the chromosomal aberration or abnormalities. These flaws may hinder the pig's ability to perform its duties or perhaps result in its demise. Some genetic mutations are completely benign or harmless. Others can affect the animal's phenotype, resulting to abnormal development. Also, aberrations can affect the reproduction process, as litters may not reach full term or it may reduce the prolificacy of the mother animal. Because congenital malformations are frequently severe and apparent, producers have always thought that all anomalies are hereditary in nature, but the normal prenatal growth of the pig may be affected by virus infections, nutritional deficits, and consumption of specific medications, toxins, and pesticides during pregnancy. Abnormalities from the normal growth and development may disrupt internal environment, resulting in a change in the number of chromosomes from the wild-type number, or the normal. In this case, the normal number of chromosomes in pigs is 38. In this part of the topic, we will discuss how the change in this number of chromosomes will affect a pig. These types of birth defects occurs when there is a different number of chromosomes in the cells of the body from what is usually found. The loss and or gain of a whole chromosome or chromosomes comes in euploidy or aneuploidy. Euploidy affects all sets of the chromosomes, while aneuploidy affects only one set of chromosomes. And remember that each set of chromosomes contains two chromosomes, and this affects meiosis, gametogenesis, and the viability of zygotes and embryos, as what I have said earlier, that when fertilization occurs and the gametes has abnormal amount of chromosomes, it could result to zygotes or embryos not reaching its full term. Consider the case report on the phenotype of a pig with monosomy X resembling a Turner syndrome symptoms by Kazuhiro et al. Monosomy is a type of aneuploidy. Remember that aneuploidy affects only a single pair of chromosomes. If pigs has 38, then it has 19 pairs of chromosomes. And in a monosomy, one of the chromosome is lacking a pair, resulting to a reduction of chromosome number to 37. This results to short stature, microglia, skeletal abnormalities in the limbs, no estrous cycles in the female even after reaching sexual maturity, no ovarian endocrine activity. Now, let's move on with a different kind of chromosomal aberration which now affects the structure of the chromosome. The chromosomes of the domestic pig or Suscrofa domesticus are known to be prone to reciprocal chromosome translocations and other balanced chromosome rearrangements with concomitant fertility impairments of carriers. A balanced chromosomal translocation is a condition in which a part of a chromosome has broken off and reattached in another location. In other words, it means that sections of two chromosomes have switched places. There is no acquisition or loss of genetic information, hence there is no phenotypic effect. But recent studies have shown that it has correlation with reproductive problems and one of the clinical signs of this aberration is hypoprolificacy. Hypo means below the normal and prolificacy means the amount of litters or young produced. Basically, if a certain breed produces 12 viable litters normally, a hypoprolific sow or a mother pig will only be able to produce 5 to 7 liters. A similar result showed in a study of De Souza et al. Within Australian boars. Out of 94 boars, 6 boars were identified with 4 balanced chromosomal rearrangements. And within those 4 boars, it showed a reduction of litter size from between 25 to 75%. Now, before we discuss how these aberrations are identified, let me first explain why it is important to spot them. First, these aberrations are hereditary and can be passed on along the pedigree. Such clinical signs like hypoprolificacy and abnormalities affects animal production and economics. Ideally, farms should have less production input and higher production outputs. If generations of pigs become carrier of these mutations, the litter size will eventually go low as years go by. Now, one of the things intensive farms can do to spot a carrier pig is to check for any clinical signs and perform routine karyotype screening of animals qualified for reproduction. A karyotype is an individual's complete set of chromosomes. The term may also refer to a laboratory-produced image of a person or animal's chromosomes isolated from an individual cell and arranged in numerical order. In this way, aberrations such as monosomy and translocations will be seen. Unfortunately, once a pig is an identified carrier, it will be isolated, culled, or etc. from the rest to avoid producing litters that may be potentially carrier of the mutation.